In the second part of today's lecture, we heard how the CRISPR system, which of course is part of a, if you like, an adaptive immune system of bacteria, we saw how we can adapt this using the tools of molecular biology to generate specific genetic changes in genomic DNA of human cell lines. This CRISPR system is one of the greatest advances in molecular and cellular biology over the past 5 to 10 years. It can be, of course, used to delete enhancers. And we've heard a great deal about enhancers in this unit, how many of the GWAS that have been carried out have identified enhancers which drive the tissue-specific expression of genes that are associated with specific phenotypes, whether it is obesity, schizophrenia, or asthma, or type 2 diabetes. There is an underlying theme of common polymorphisms being found in these enhancers and subtly changing the transcription of the target gene. In this particular figure here, it's a reminder that we've already seen this in the gene FTO. There is an intronic enhancer, approximately 500,000 base pairs away from its target gene called IRX3. You could imagine the use of CRISPR to delete this enhancer using the Cas9 CRISPR system. So what we're going to look at now is a study done looking at what's called Parkinson-associated risk variant in distal enhancer of alpha-synuclein modulates target gene expression. So before we go any further, let's just unpack what this title is talking about. So Parkinson's disease, of course, is what it's talking about. And this is another neurological disease. We're looking at risk variants. So this is probably a fairly low penetrance variant that we're talking about, perhaps identified in a genome-wide association study. We're looking at a distal enhancer. Distal means that it is far away from one end of the gene. The gene itself is called, or it encodes a protein called alpha-synuclein, and you can see that it targets gene expression. So what we're looking at is a variant in an enhancer that controls gene expression of alpha-synuclein, and it's important in understanding some aspect of the phenotype of Parkinson's disease. So as I've said before, GWAS have identified DNA loci associated with many illnesses, including Parkinson's disease. These GWAS often identify enhancers that regulate gene transcription in a cell-specific manner. Multiple genetic variants in the gene encoding alpha-synuclein, or SNCA, are associated with an increased lifetime risk of Parkinson's disease. The SNP variants in this gene SNCA that are most strongly associated with sporadic Parkinson's disease increase lifetime risk by around 30%. Now that's 30% against the normal healthy population. Significantly, more than half of the world's population carries these risk-associated variants. And that really illustrates something we've talked a lot about in this course, which is that just because you have the variant doesn't mean you develop the disease. We know now that these diseases, from a genetic point of view, are incredibly complex and often with a strong environmental input as well. So understanding the effects of these genetic variants is important in the context of public health. So Parkinson's disease then, it's a result of what are called a loss of dopamine-producing brain cells or neurons. These dopaminergic neurons play an important role in a number of different brain functions, including the control of voluntary movement, as well as a range of different behavioral processes such as mood, reward, addiction, and stress. There are 4 symptoms of Parkinson's disease. These are tremors, trembling in the arms and legs, rigidity and stiffness in the limbs and trunk, and a slowness of movement. There is also an impaired balance and coordination. If we look at a brain and the dopamine pathways, in pathway 1, which is called the nigrostriatal pathway, this is where we see death of neurons linked to Parkinson's disease. And it is this pathway here that is linked to loss of motor control that typifies Parkinson's disease. There are two other pathways which impact on reward, desire, and other behaviors. If we look here, for example, pathway 2, for example, if we see any defect in there, we start to see hallucinations and phenotypes associated with schizophrenia. Alpha-synuclein is a member of a larger family of these synuclein proteins. It is abundantly expressed in the brain, and it's thought to integrate presynaptic signaling and membrane trafficking. In other words, it plays a role in the transmission of signals between neurons. Defects in the gene SNCA have been implicated in the pathogenesis of Parkinson's disease. Significantly, synuclein A peptides are a major component of the amyloid plaques in the brains of Alzheimer's disease patients. Coding mutations and genomic multiplications or gene copy variants of this gene cause familial Parkinson's disease, whereas rare variants and common variants, such as we'll talk about today, are associated with the sporadic form of the disease. In a way, this is somewhat reminiscent of Alzheimer's disease, where we saw pathways beginning with the APP protein, the processing by the gamma secretase, leading to the processing, clearance, and so on of the beta peptides. These are the familial form of Alzheimer's disease, a mutation in the APP protein and the gamma secretase, for example, and the more common variants which impact on the clearance of the resulting beta peptides. Synuclein also has familial forms of the disease, which is these rare high penetrance mutations, as well as more sporadic forms of the disease, which are linked back to these common alleles. As we saw, up to 30% of the population may carry a risk allele for Parkinson's. Given that SNCA peptides are also a major component of amyloid plaques, it's not surprising that it is the overproduction of synuclein that is associated with toxicity and the onset of Parkinson's disease. What we see here, again, a fairly complicated looking diagram, so let's take our time to look at it carefully. This is showing the location of SNPs that have been identified in genome-wide association studies that are associated with sporadic Parkinson's disease. So what we see is, here is our chromosome, and it's chromosome 4, and this little red dash that we see here indicates the location of the gene SNCA that encodes the protein alpha-synuclein. There is an enhancer located in the alpha-synuclein gene, and it's in intron 4, which is here, as well as this, what is called a 3' UTR or untranslated region. What we see in this list running down here are different parts of the brain, substantia nigra, hippocampus, etc., all the way down into fetal brain. And they've looked at epigenetic marks in this region of the gene. Remember, we can use epigenetic marks or modification of our histones, as well as the opening up of chromatin to indicate the location of an active enhancer. If we just focus on the green and blue boxed areas, this is the substantia nigra, which is this area where we see the loss of dopaminergic neurons associated with Parkinson's disease. These peaks show that there is a high level of histone 3, lysine 4 monomethylation, which is a marker for an active enhancer. We also see very robust peaks of histone lysine 27 acetylation. You remember acetylation, it opens up the chromatin. And down here, finally, this is the active marks of open chromatin. So we're seeing that this region of the gene is carries epigenetic marks that suggests it is actually an active enhancer. What we are in effect looking at, of course, is an EQTL, or an expression quantitative trait locus. You remember, if we have two copies of a gene, one copy of the gene carries perhaps an A allele in its enhancer, whereas the homologous copy of this gene carries a T allele in the enhancer. This results in differences in expression from the AA of high expression to the TT of low expression and the AT somewhere intermediate. This change in expression that is associated with different alleles is called allelic imbalance, and it is a hallmark of an expression quantitative trait locus. So what did these authors do? They wanted to test, first of all, whether this region of intron 4 in the alpha-synuclein gene actually included an enhancer. The second question they were interested in asking is, what is the key nucleotide, the key allele that is required for the enhancer activity associated with Parkinson's disease? In this diagram is shown a series of experiments that were done by the authors to test the function and role of the enhancer in the intron of the alpha-synuclein gene. What we see at the top are two SNPs, shown in red, that have been identified in genome-wide association studies and which map to the putative enhancer. We see here that there are two copies of the alpha-synuclein gene, and we're looking at a particular cell line derived from a human brain tissue. And this cell line has either an AT haplotype, or on the second copy of the gene, it has a GT genotype. In the first experiments, the authors used the CRISPR nickase mutants to delete the enhancer from both copies of the gene, and this is called delta E4, delta E4. In the second experiment, they performed what is called an insertion. In other words, they now wanted to study the effects of specific haplotypes or allele combinations on the function of this enhancer. So now they've created a heterozygous cell line. One copy of the gene lacks the enhancer, and in this case we see an AT combination. In a second cell line, again, the deletion of the enhancer, and now we're testing the GC combination. The third is an AC, and the fourth is a GT. So in other words, what the authors are now asking is, what is important, the A allele, the G allele, or the T allele? And of course, the use of a C allele will allow them to test that. In this figure is shown the results of their experiments. We see here the names of the cell lines that I've just described, delta E4, and so on. What we can see is that deletion of the enhancers results in a low level of expression of the alpha-synuclein gene, as does the haplotype AT and AC, shown here. However, as soon as we introduce a G allele, what we see an increase in the expression of alpha-synuclein. This led the authors to conclude that it is the G allele in this enhancer in intron 4 of the alpha-synuclein gene that is associated with increased expression of alpha-synuclein. The function of this particular enhancer works in this way. So here we have our A allele, and down the bottom we have our G allele. The A allele is a protective allele. It results in efficient binding of a transcription factor that represses the expression of alpha-synuclein. The repression of alpha-synuclein reduces Parkinson's disease risk. However, a G allele prevents the binding of the transcription factor, allowing enhanced transcription of the alpha-synuclein gene. The transcription of alpha-synuclein increases Parkinson's disease risk. So what we've seen is that there is alternate binding of a transcription factor. If it's an A allele, the transcription factor will bind and it will suppress the activity of alpha-synuclein. If it's a G allele, the transcription factor will not bind and we will see an active distal enhancer. The alpha-synuclein will be expressed at a relatively high level. This illustrates that we have transcription factors, protein-DNA interactions, that can either activate or repress gene transcription. And in this case here, we have a transcription factor whose function is to repress gene transcription. So the alternate allele prevents the binding of a repressive transcription factor, allowing transcription of the gene. The function of alpha-synuclein is not well understood, but it is known that the presence of a mutant form of alpha-synuclein, which is the A53T mutation in familial Parkinson's disease, or under conditions where its expression levels are increased, becomes a toxic protein that is associated with increased rate of neuronal cell death. So what these authors have done is that they've used CRISPR to delete an enhancer in the alpha-synuclein gene. They've seen increased levels of alpha-synuclein expression in these cultured cells, and that the risk allele, the G allele, is controlling the activity of this enhancer. This CRISPR is an incredibly powerful technique for testing whether or not an enhancer is active, and also for identifying the true functional allele within that enhancer. Of course, we can increase that to thinking about the use of CRISPR in iPSCs, or induced pluripotent stem cells. You could imagine that you could generate a cell line from a patient, for example, with familial Alzheimer's disease, and you could start using CRISPR to modify other loci in the cell and to test the interaction between these common variants and rarer high penetration mutations. So CRISPR, together with induced pluripotent stem cells, are providing biologists and geneticists with the opportunity to study, under much more realistic conditions, interaction between mutations under defined culture conditions and their effect on cellular phenotype. Of course, it also provides an opportunity for the testing of drugs that may modify the phenotype of a neuron, for example, from Alzheimer's disease or from Parkinson's disease. CRISPR-Cas9, as we've seen, the combination of this CRISPR-Cas9 nuclease and target-specific RNA that we call a guide RNA induces double-stranded DNA breaks, or in the nickase mutant model, single-stranded DNA nicks. This is repaired by DNA repair pathways. And we can apply this to mammalian cells to introduce deletions and mutations. And this, of course, is done in cultured cell lines. Which raises the question, can we, should we, do we edit the genome of a human embryo? This slide here shows that in different places in the world, there are different approaches to this. For example, in Europe and Australia and other parts of, and Canada, for example, there is a complete ban on the use of CRISPR on embryos. In other parts of the world, for example, here in the United States, it does not allow the use of federal funds to modify human embryos, but there are no outright genome editing bans. So you can see that generally, at least in the Western world, it is highly restrictive, highly controlled, and really not possible to apply this to humans. And of course, it raises huge ethical questions if anyone were to consider it. However, about just over 1 year ago, this bit of news came to light. And what they're doing here is that scientists in London have been granted permission to edit the genomes of human embryos for research. Now, you might think this goes against what I've just told you, but actually what they've done is that they've gained permission to work on embryos or blastocysts from IVF during the first few days after fertilization. Their ethical approval said that they must stop the experiments after 7 days, after which the embryos will be destroyed. The genetic modifications are thought to be useful in understanding how to develop treatments for infertility, but will not themselves form the basis of a therapy. Now, of course, even in this use of IVF embryos for this research purpose is, for many, involves very, very difficult ethical questions. Gene editing, editing of the human genome, continues to appear in the news, and it is continually argued and discussed and tightly regulated.